To kick us off today, I'd like to welcome Sandhya Prabhakaran. She's a research scientist at Florida's Moffitt Cancer Center. She's here to discuss her work on single-cell RNA sequence data and imaging. Throughout the event, you can view presentation decks within the console resources icon. Go ahead and start dropping your questions in the Q&A panel and we'll answer them at the end. With that, I'll turn it over to you, Sandhya.
Hello, everybody. My name is Sandhya Prabhakaran, and I'm a research scientist at the Integrated Mathematical Oncology Department, Moffitt Cancer Center, Florida. The work that I'll be presenting today was done while in the lab of Dr. Dana Pierre, Sloan Kettering Institute at Memorial Sloan Kettering Cancer Center, New York City. So I have the extreme pleasure of presenting the machine learning and computational biology, the work that I've been doing for quite some time, using single-cell RNA-seq data and spatial transcriptomic images. So in the first part, I'll explain about my work dealing with single-cell RNA-seq data, and I'm the lead author of a method called BISCIT, which stands for Bayesian Inference for Single-Cell Clustering and Imputing, which clusters and normalizes single-cell RNA-seq data. So what is single-cell RNA-seq data? Here you can actually take samples from tissues which are either normal or tumor, and you can isolate cells and sequence the individual cells to get information such as what genes are present in a cell. This is a very heavily involved biological process, but at the end of the day, what you get is a count matrix that you see here, where you can quantify the number of cells that is present in the sample and the gene makeup in each cell. So for example, a count matrix like this, and you can now use this for any sort of downstream analysis such as clustering or graphical network analysis. And what you see here is a cluster, which is now a T-SNP plot where every dot is a cell and colored by the cell type that is present in that particular tissue. So what is the importance of single-cell RNA-seq data? This actually helps you get the transcriptome-wide gene expression measurements along a cell resolution, from where you can identify cell-cell clusters, the arrangement of population of cells according to different hierarchies, and the identification of cell transitions between states. It also gives a much clearer view of the dynamics of tissue and organism development and on structures that are within the cell population that were so far perceived as homogeneous. Single-cell RNA-seq was a method that was highlighted as a methods of the year 2013 by Nature Methods. And now this is a burgeoning field with so many other spatial multiomics techniques as well coming into play. So a quick recap of what are the problems when you sequence single-cell RNA data is that definitely you have biological errors that can come through the techniques, wherein you're only able to sample certain portions of mRNA or transcripts as they are called, and now you have missing information. There are amplification differences when the cells that you need in the tissue are, let's say, very sparsely populated, and then the cell type specific capture rates can be different. Sometimes there are technical variation that can then be mistaken for biological heterogeneity. So a simple example is over here, where you have, in the particular single-cell RNA-seq matrix, you have the count matrix that you obtain where rows are genes and the columns are the cells, and you have three clusters, let's say. If everything were perfect and there were no noise, you would get three different clusters like you see here. It's a 2D projection. Every dot is a cell. But ideally, in reality, this is what you would get. Because of missing data and noise, the T-SNP projection is very random. Now, different approaches to clean this up is first by doing normalization and then clustering, and then you do downstream analysis. But the so-called missing data, known as dropouts, are not resolved, like zeros still remain as zeros, and you're removing any biological stochasticity that might be there, which is also not very helpful. So we developed this algorithm called BISCIT, but where there are two different problems that are being solved. The first one is that of imputing. So for example, if you look at the tissue that I've shown before, and you look at this particular cluster here, and you don't know how to impute the information, you don't know what genes are present here, but looking at the neighbors, you can say that perhaps the cell might have something to do with gene A. The squiggly lines here are genes. And like I said, you can assign gene A to this cell. What if don't have any such neighbors having a good expression of gene A? In this case, you would look at co-expression. So the neighboring cells have A and B genes, and the cell in consideration has gene B. Therefore, you can now assign gene A to cell this cell. So this is how you would impute the data. And another problem was that of normalization. So you want to normalize the data to keep the library size, which is nothing but the number of mRNAs per cell, the same. So one of the examples that is normally done in the field is that of global normalization. So let's take the example of 3 different cell types. You have macrophages, which are bigger cells, lymphocytes that are tiny, and neutrophils that are somewhere in the middle. Now, the library size for each of these would vary depending on the amount of mRNA per cell type. But you can see that housekeeping genes, for example, these are genes that are supposed to be there in that particular cell. Their contents also vary. So once you do a global normalization, you actually change the library size variation in these different cell types. But you can see that the dropout isn't resolved. And sometimes you have spurious expression that comes in. So these are all leading to biased results if you use them. So one of the key ideas we had for BiSKIT was to use a different normalization for each cell type in such a way that after you normalize them, you get the library size the same. Like if you look at the upper bar, they're all the same, but then these are now cell type specific. But this is now a chicken and egg problem. How would you do this? Because you do not know what the cell types are. And therefore, we came up with this iterative procedure where you take the data, build the count matrix, cluster it, and then impute and normalize. So for this, we've used a hierarchical Bayesian mixture model. And probably wouldn't go into too much of the math, but I left this here for anyone who's interested. We assume that each gene here, which is a row, is a mixture of log normal models and having a representation for each of the 3 clusters that we saw. If you take 2 genes, now you have a pairwise model coming up. So genes themselves can be modeled as a mixture of multivariate log normals, and this is a pictorial depiction of how these 3 different clusters exist. And now we have something that is added to these to these clusters, which are cell specific. So this is a ginormous hierarchical mixture model, which then has to be solved. And then you can get the different clusters that are also cell type specific. So I've left this in for reference, but I won't go into this in detail for now. And like I mentioned before, this is an iterative procedure where you build the count matrix, cluster it, sample the cluster specific parameters, sample the hyperparameters so that you can use it with other data, and sample the technical variation that belongs to each cell, cluster, and this goes on. So this is a variation of the Gibbs iterations in case anyone is interested. I've left the reference of the paper here in case anyone is interested to go through it. This is again another pictorial representation of what really happens in BiSKIT. At the end of the day, we are able to normalize each cell based on its cell type, so macrophages, neutrophils, and lymphocytes, and retaining the housekeeping genes in the same number as required. This is again an example of the performance of BiSKIT on actual data. So these are the cell types or the clusters. Every point is a cell and every color is a cell type. You can actually identify that there are 7 different colors, so 7 different cell types taken from a publicly available data set. And this was BiSKIT's performance on that data set, which means that we have benchmarked this properly. And this is the application of BiSKIT on a real-world data set. These are taken from 4 different patients, breast cancer patients. There are a total of 9,000 cells. If you do global normalization, you can see that the clusters tend to show patient biases. Each color is a different patient, but if you use BiSKIT, you can kind of see a defined structure with corrected patient biases. All right, so that was the work on single-cell RNA-seq. I'm also particularly interested in working with spatial transcriptomics images, and I'm also the lead author for a new work that I've been working on, and it's currently in bioRxiv, called SPARKLE, which means spatial reassignment of spots to cells using maximum likelihood estimation. So I'll begin with an example of what a spatial transcriptomic image looks like. An example would be MERFISH. So this is one field of view where what you can see here in small circles are the cells. And then, of course, you would like to do cell segmentation. So the red lines indicate the cell segment. And you can actually see small dots in between, all across. And every dot is an mRNA spot or an mRNA transcript. And the color there is indicating which mRNA belongs to which gene. So here you would find 140 different colors, meaning 140 different genes, but so many different mRNAs that belong to those 140 genes. So this is a very intense data set, and there are around 400 different fields of view for us to work with. And what you see here is just a name of one particular gene that's available in this data set. Now, why is spatial transcriptomic imaging so important? Again, like I said, this is used to assign different cell types because now you also have the mRNA locations, which is a piece of information that you use in single-cell RNA-seq data. And now you're also able to know where exactly those mRNA molecules are. This was recently named as the Method of the Year 2020 by Nature Methods and was also identified as one of the 11 critical grand challenges by Genome Biology. There are many large consortia like Human Cell Atlas, the Allen Brain Institute, the Human Tumor Atlas Network that are undertaking a huge data generation projects using single-cell transcriptomics imaging. I've shown you MERFISH, and these are other examples of spatial transcriptomics images out there. What was my particular research question? So this is an example of a MERFISH image that I had shown in the previous slide. The green fluorescence that you see here is again a cell nuclei. What I've plotted in the second panel here, the mRNA transcripts that both belong within the cell and outside the cell. So the ones that are within the cell are marked in blue, and the ones that are outside are marked in pink crosses. So like you can see, there's so many pink crosses outside, which currently in downstream analysis are discarded, simply because you do not know which cell they belong to. And panel C, you can actually see the spatial diversity of transcripts, which are shown as dots that are belonging to within the cell. So here you see a bunch of dots, and then outside you see different colored crosses. So these are transcripts that do not belong to any cell at the moment. So in Sparkle, what we were doing is we were trying to recover the pink crosses that were outside the cells and assigning them to cells. So like you can see, this is a huge computationally expensive problem. So we do not assume any uniform distribution of mRNA transcripts within the cell. And like you can see, they're very far and spread out. And there's a large number of transcripts in the cell that allows you to use a multivariate Gaussian distribution for clustering. What was required was a multiplex FISH images. We needed to start with some sort of a prior, and that was the cell segmentation. And we needed to know which of the 140 genes belonged in this data so that the mRNAs could be mapped to it. And the outputs were improved transcript assignments to cells, a better count matrix, a better delineation of the cell boundaries, and improved cluster assignments. So some of the principles here, like I said, we generate a count matrix, and you can apply any of your clustering methods that you like. We build what is known as a mock cell for dangling transcript. And in this image here, this is again one of the ones that I've shown in panel C before. I have a pink cross here, which is actually a dangling transcript that we call a dangling mRNA. And we build a circular window around it and quantify the number of mRNA that are around it. So we're building a mock cell that is centered at the dangling transcript. And all the details of this is present in the paper. Once we have the mock cell, you can also weight the mock cell. And then you use the maximum likelihood estimation to assign each of these dangling transcripts to nearby cells, update the count matrix, cluster, and iterate. In our observations, just 3 of those iterations were enough to recover a lot of these dangling transcripts to the cells. This is a flowchart in case anyone is interested for a take-home message. Again, an image of a MERFISH, a zoomed-in image here, which shows the different kinds of cell types. Again, another zoomed-in version here showing the different transcripts that are both inside and outside the cell. Here we show an example image of what we obtain after Sparkle has run. So every line here shows the assignment of a transcript to a cell, and the color is based on the cell type. This then gets updated to the count matrix, you can cluster, and the process continues. This is again some results that we see. This is from the first iteration, this is the second, and this is the third. Panel C actually shows all of the assignments that we've had so far. These are recovery statistics of the transcripts when we compare with single-cell RNA-seq, MERFISH, and MERFISH with SPARKLE. And here's a pie chart which says all the areas in dark pink is what is being recovered from SPARKLE. And this is now showing how good using the data from post-SPARKLE data, post-SPARKLE would be, and how much of information we've recovered. And this again shows it in with respect to canonical genes for each cell type. Some of the future work would be to write this in a language that is more effective like Java, to use a more Bayesian version of a posterior inference technique, and to improve cell type boundaries, which is now a question for the field. In conclusion, I'd like to say that I've worked on single-cell RNA-seq and I've contributed to the development of BiSKIT. I've also contributed to single-cell spatial transcriptomics through SPARKLE. And some of the open problems in the field are the need of having more computational models to handle big data, combining all this big multimodal data that comes from images, RNA-seq, ATAC-seq, for example. And another is of understanding the biological mechanisms that give rise to these data patterns. So if anyone has any questions or would like to have a chat, please find my email and my Twitter handle here. And thank you so much for listening.
Yeah, I just have a question.
Perfect. Yeah, we've had one come in for you. Let me pull up. So one person in the audience asked, how do you relate to TF using scRNA sequence?
Right. So this is actually a very great question, and there has been work that's done in this direction where you connect single-cell RNA-seq data with epigenetic data. And one of the tools I can think about is termed Maestro. So what they do is you basically look at the transcription factor peaks, find their